Welcome back, everyone. Today we'll be talking about Chapter 4, which is perhaps one of the most important chapters of the course, since I like to think of this as the North Star. This is really going to be a lot of the information that you're going to need to understand as we start to talk about specific diseases and start looking at specific classifications of microbes to understand how they're placed into those different classifications, but just to understand the anatomical and physiological structures that allow microorganisms to function. So, this is really just a very important chapter for you to thoroughly understand. And one of the things that we're going to do today is we're going to start off talking about some of the characteristics of living things. In earlier chapters, back in chapter 1, we discussed the difference between our prokaryotic and eukaryotic microorganisms. We said that prokaryotes had a pre-nucleus form, meaning that they did not possess a nucleus or other compartmentalized membrane-bound organelles. Eukaryotes, on the other hand, are much more compartmentalized. You can think of it as kind of like a blueprint on a map for a house, where you would see, all of the different rooms and the closets within the rooms. Eukaryotes possess a nucleus, but then they also have all these other compartmentalized, what we call organelles, that execute a particular function within the cell. So we talk from there about things like the viruses, which we said are comprised of proteins and nucleic acid, but are inert and are acellular, meaning that they require a host cell for all of their replication to occur. So we're going to make a difference today, and we're going to speak specifically today in Chapter 4 about the prokaryotes and the eukaryotes in terms of their characteristics. So viruses, viroids, prions, which we'll talk about later in the course, do not fall into this category. So without further ado, let's kick it off. All right, and let me increase my screen here so everyone can see it. So we're going to talk first about characteristics of cells. So all living things, whether you are a single cellular, which we call a unicellular, or multicellular, meaning that you're comprised of multiple cell types that all carry out a different function. We as humans are multicellular. We have cardiac cells, we have epithelial cells, and all those cells execute a different function and purpose within the body. Things like a yeast are considered unicellular. They only have one particular cell type that comprises the organism. All living things have a basic shape, whether it's spherical or cylindrical. We'll talk about particular shapes. And when we're talking about bacteria, bacteria can be what they call cocci, and those are spherical, or they can be bacillus, which has this cylindrical shape. They have some type of an internal content. Most of the internal makeup of a cell is this gelatinous material, which is the majority of its water, about 70% of it is water, and it's referred to as the cytoplasm. And this cytoplasm is all kept together by a external membrane. So there's a membrane that surrounds that cytoplasm, usually comprised of a material called phospholipids. So these phospholipids have properties to them that part of the molecule, the head of the molecule, is water loving. We call that hydrophilic. And the tail end, which faces in towards the center of the cell, is what we call hydrophobic, these kind of long tails. Because they have a high lipid property to them, they are water faring. So they are hydrophobic and they want to stay away from the external environment of the cell. The cytoplasm is really important because that's where most of the chemical processes occur within these cells. And then finally, not only do they have a shape and an internal structure, but they also have a particular type of nucleic acid which carries the blueprint of their life to them. And this is usually in the form of a DNA chromosome. And that DNA chromosome is wound really tightly. So it's this condensed, really organized piece of genetic material. These cells also possess ribosomes, which are responsible for protein synthesis. So we need a way for us to be able to carry on our genetic code through our DNA. But then we also need ribosomes to execute the functionality of creating proteins for the cell. And as we already mentioned, we have two basic cell types. We have the eukaryotes, which include things like the algae, the fungi, the protists, and the helminths when we're talking about microbes. And we have the prokaryotic, and those we mentioned are either the bacteria or the archaea. And remember that the archaea are those extreme, they live in the extreme environments, and they differ from the bacteria in terms of the molecule that makes up their cell walls. They have a material known as pseudomurine, which is different from the peptidoglycan that makes up bacterial cell walls. Eukaryotic cells, as we mentioned, because they're different from prokaryotes, remember eukaryote means true nucleus, they contain not only the nucleus that possesses all of the DNA, but they also have these compartmentalized organelles that execute different functions within the cells. So cells are having to go through many, many, many different types of functions throughout their lives. So they have to be able to, obviously, at the top we see here, they have to be able to copy their DNA to pass on to daughter cells. As the cells grow and divide, they need to make sure that that genetic material is also copied effectively and efficiently and passed on inside the new daughter cells. So the copying of this genetic material ties very nicely here to the growth and development. Cells, we're going to see, also need to be able to respond to the external environment. They're going to be constantly moving things, as we see at the bottom here at transport. They're going to have to be moving small molecules in and out of the cell, whether it's a waste product that needs to leave or a nutrient that needs to enter. They're going to need to constantly pass materials through these different types of channels and carriers to get them in and out of the cell. Cells also need to contain those proteins, these things that we call enzymes. So one of the questions is, how do you know if you're dealing with an enzyme? So when you're looking at a protein, how do we know if it's an enzyme? Well, the easiest hint for you is an enzyme is always going to end with an ASE suffix. So the end of the word will always be an ASE. So for instance, something like permease or ligase, all of those end in ASE, which denotes that they are enzymes. So that's just a little trick for you to carry on throughout your science classes, throughout your eventual clinical programs, to understand what that ASE ending represents. So these enzymes have a job to catalyze all of these different metabolic reactions within the cells. So it's really important for them to be able to eventually take in a what we call a substrate or reactant and convert it into a product. And then obviously reproduction is important. If you can't go on and create new cells, then you are going to fail to exist. So we already talked about the genome, or the area where all of the nucleic acid is found. The DNA is packed in these chromosomes and is found in this region known as the genome. We talk about two different types of reproduction. Sexual reproduction, which obviously means that you have the combination of an egg and sperm, or asexual, which happens in the absence of egg and sperm, and it's usually through some type of a process like a cell division, where one cell divides into two new cells. We've talked about growth and development, metabolism, Movement is really important as well. We're going to talk later about what's known as taxis, or the ability to be able to respond to different stimuli, whether that be light or chemicals. We often refer to phototaxis as a response to light, or chemotaxis as a response to chemicals. We also have the ability for the cell to be able to protect itself, to be able to support itself. The cell wall on bacterial cells is critical to protecting the cell from damage, but also large amounts of osmotic stress. So, as different materials move in and out of the cell can either expand or contract. And as the cell expands and gets larger, we don't want it to burst like a balloon. So the cell wall protects the bacterial cell from breaking under high levels of stress. So here's just an overview structure of a bacterial cell. And as you can see, it's very simple. There is not really a ton going on inside the cell. And today, what we're going to do is we're going to start to unpack some of these particular segments and pieces that comprise these truly unique cells. So on the outside of the cell, we're going to start on this kind of brown layer that you see out here, this external layer. We're going to talk about these particular structures. So there's two different functions of the appendages that are found on the external parts of a bacterial cell. And one of the functions is for motility or movement. So we're going to talk about either flagella, which are these kind of long whip-like tails that you see back here that extend off of one of the sides of the cell. We're going to see that they can either be polar, meaning that they come off of one end, or there's cases where flagella can be found over the surface of the entire cell. So we have some different arrangements of these flagella, which again are turned in a particular direction to propel the cell forward. In the case of the spirochetes, okay, the spirochetes are kind of these wavy appearance bacteria. The flagella actually extends inside the top portion of this membrane on the outside of the cell. And we call this a periplasmic flagella, meaning that it's kind of embedded within the membrane. And it gives the spirochete this kind of twisting motion as this periplasmic flagella, or what we know as an axial filament. The appendages can also be used for attachment or channels to move things in and out of the cell. So we have the Fimbriae. Be careful. Flagella is different from Fimbriae. Fimbriae are used solely to adhere or attach to surfaces. Pili are these channels that allow for the movement of genetic material. So we talk a lot about a process in bacteria known as conjugation, which is essentially bacterial reproduction, where a donor can actually pass genetic material on to a recipient cell through this channel known as a pili. And then we also have this layer that forms on the outside of the cell, which is known as the Glycocalyx. That's this kind of surface coating. And in a few moments, we're going to see that there's actually two different types of Glycocalyx. So when we talk about the flagella, there's three parts to the flagella. There's the actual filament, which is this long strand, this wavy strand that comes off of the cell. We have the hook, which is what kind of ties it and helps it stay on to the cell attached to this basal body, which is a stack of rings that anchors the flagella into the cell wall. So here's kind of an overview for you. Here's the filament that we were talking about. Here's the hook, which is the beginning of this anchor mechanism, which ties in down here. And this, again, this basal body structure is what anchors it right into the cell wall, which you see down here, or cell membrane, excuse me. The flagella, again, rotates in a 360-degree direction. And depending on the way that it rotates, so if it goes either clockwise or counterclockwise, so in this case, this is going clockwise, the cell is going to run or move forward in a straight line. If it rotates the opposite direction and it goes counterclockwise, the cell is going to tumble or do this kind of somersault where it goes head and it rolls kind of like you would if you were somersaulting. We mentioned that there's different ways that the flagella can be arranged over the surface of the cell. So the simplest one is known as Monotrichus, and this is where we have a single flagella that's attached to one end or one pole of the cell. Lophotrichus, on the other hand, is where we have a small bunch of flagella that are coming from one pole of the cell. So monotrichus would be just one, think mono meaning one. And then lophotrichus would be the next level up where we have a tuft or group of flagella coming from the same pole of the cell. Amphitrichus means that we have flagella coming from both poles of the cell. So we have it at this pole, and we also have flagella at this pole. And then peritrichus, you'll notice that the entire surface of this cell is covered with flagella. We have it at the poles, but then we also have it along the actual outer external layer of the cell as well. So as you go forward, I want you to think about which one of these arrangements do you think would be most productive for bacterial movement? And if you are, as you start to post, if people put this as a topic in the discussion board, feel free to comment and let us know what you think. We also want to talk about how flagella respond. So we already mentioned that they can move their flagella in a clockwise or counterclockwise direction, depending on if they want to run or tumble. But why would they choose to run versus tumble? And that's because of the stimuli that they're exposed to. So if they're exposed to a chemical stimuli, this is known as chemotaxis, and it can be either a positive or a negative chemotaxis. So what does that mean to be positive or negative? Well, if I was to right now hold out a suitcase with $10,000 in it, everybody's going to come running forward to try to figure out how they can acquire that money. But if I, put on a Halloween mask and ran at somebody, that's a negative chemotaxis. So people are going to want to run away from that. You're going to want to get away from that if it's something that is you're fearful of. Well, the bacteria respond in the same way. If they're exposed to a chemical nutrient, they're going to want to move towards it, or a positive chemotaxis. If it's something that's a toxic waste product, that's going to solicit a negative chemotaxis where they're going to want to move away from it. Light stimuli, photo always means light when we're in biology. So a phototaxis is a response to light. And we already mentioned a counterclockwise rotation results in this nice, smooth, what we call run. Clockwise results in this kind of somersault pattern, which we call a tumble. And we already mentioned things like the spirochetes can have this internal flagella, which is enclosed within the outer membrane structure of the cell. We call it that periplasmic space. And it gives this kind of twisting or twitching motion due to the way that this particular flagella moves and contracts within that space. And this is important with the spirochetes, the bacteria that are kind of this wavy shape. So we've kind of already talked about this. The Fimbriae are these hair-like bristles that are located on the external portion of the cell. And again, they're Sole job is for adhesion and adherence to other cells and other surfaces. We talked about the villi, which are these tubular structures found only in gram-negative cells, and we'll talk about what gram-negative means in a few. But they join cells together for that DNA transfer. We call that process conjugation, where the donor transfers its genetic material through these pili from a donor cell to a recipient cell. And we also mentioned the glycocalyx. And the glycocalyx is this outer coating that's consisting of sugar molecules. And there's two different types. We have this really, you'll see this one here. It's this very loosely disorganized layer of coating on the outside of the cell, which we call the slime layer. We can also have this much more defined, neat, and concise slime coating, which we call the capsule. And it's highly organized, very tightly attached to the cell. Whereas that we said the slime layer is this highly disorganized and very loose layer on the outside. The glycocalyx has several functions to it. So it's going to protect the cell from being dehydrated, losing its water, or losing nutrients. It prevents something known as phagocytosis. So we have white blood cells as part of our immune system, phagocytes and macrophages, which circulate and ingest almost like a little Pac-Man. They eat up anything that's foreign. The glycocalyx can actually help the bacterial cell to avoid phagocytosis and thus allow it to be pathogenic or cause disease to the host because it's not being destroyed. And then finally, attachment. And we talk a lot about biofilms, these structures called biofilms. Biofilms are these really thin layers, these films of microbes that are growing in close proximity to each other. And they actually communicate through chemical signals. So a lot of times we think about mildew in a shower or a bathtub within a shower head. All of these nicely defined layers of microbial cells that are growing and communicating with each other. The plaque that you feel on your teeth early in the morning when you first wake up, those are all examples of biofilms. Here is a picture of a biofilm that's found on a catheter. Again, you've got all of these different cells in this biofilm that are closely adhered together, that are communicating with each other. When we talk about the next layer, so if I go back to our slide here, okay, so now we're going in and we're starting to look at the membrane structure. So the membranes that are underneath this external coating, which could be the glycocalyx, which is either that slime layer or capsule. We're talking about what's known as the cell envelope. And the cell envelope is the series of membranes that form the protective barrier to the outside of the cell. And really, what it does is it surrounds the cytoplasm, that gelatinous material in the middle of the cell. And it's composed of two basic layers. We have the cell wall and the cell membrane. And as we mentioned earlier, it's all about keeping the integrity and preventing the cell from being destroyed. We call that lysis. We don't want the cell to break open or lyse. And there are two different groups of bacteria, two primary groups of bacteria. Obviously, we have, if you want to think of it as an other category, we do have other bacteria that don't necessarily fit into these two classifications. I want to be specific with you that these two different classifications are denoted by their cell wall structure. So gram-positive bacteria have a very thick cell wall. So their cell wall is comprised of multiple layers of this material known as peptidoglycan. This is a really, really important term to understand. Peptidoglycan has been the target of bacteria as they try to create antimicrobial properties against the cell wall. They are targeting the destruction of this peptidoglycan to help themselves get in and cause disease to a host. So gram-positive bacteria have a really thick cell wall made of this material called peptidoglycan. Gram-negative bacteria, on the other hand, have a really thin peptidoglycan cell wall, so it's only a few layers thick. What we will see is that gram-negative bacteria also have an additional layer. So gram-positives have the cell wall and the cell membrane. Gram-negative bacteria have an actual outer membrane, followed by the peptidoglycan cell wall, followed by the cell membrane. So there's this extra layer that's on the outside of a gram-negative cell called the outer membrane that we also need to account for. So multiple layers, you can think of it kind of as, the sheets on your bed. Usually you have the sheet, then you have a blanket, and then you may have a comforter on top of that. All of these different layers form this cell envelope. So we talk about the structure of the cell wall, and there's a lot of chemistry to how the cell walls are formed. The cell wall itself, that peptidoglycan layer, does several different functions. First of all, it determines the cell shape. It works to prevent lysis or breaking open because if you think about it, you have water constantly moving in and out of the cell. There's also salts and sugars, and that can actually change the osmotic pressure both externally and internally. And the cell is going to either take in or push out water depending on the differences in the osmotic pressure between the internal and external environments. So the cell will either swell or shrivel depending on that. And if the cell begins to swell, we don't want it to pop like a balloon. So the cell wall actually has a very rigid structure to it that prevents this lysis or breaking open. Peptidoglycan, we mentioned, is the magic component that makes up the cell wall. And it really consists of this repeating series of these long sugar chains. We call them glycan chains. And you'll notice that there's these little dots that come off of them and they go in different directions. These are crosslinks. We call these peptide bridges, tetrapeptide bridges, that are composed of these 4 different amino acids that link one layer. So you'll notice these NAM-NAG alternating units. These layers are connected together through these tetrapeptide bridges. So if you think about making a Lego structure, if I was to take and make one long chain of Legos and I wanted to snap them together and connect them to another level of Legos, this tetrapeptide bridge here would be the piece that connects this layer to another layer below it. So we could make these cell walls several layers thick. You'll notice this one is 1, 2, 3, 4, 5 layers thick. So you can add as many different sheets to this cell wall. Remember that the gram-positives have this really thick cell wall. Gram-negatives have a much thinner cell wall. So there's only a few layers in the gram-negative cell wall. The biggest piece here is this tetrapeptide bridge, this cross-link, is what is targeted by bacterial cells, by antimicrobial treatments. So we have a class of antibiotics that target the cell wall of bacteria, and they go in and, like a pair of scissors, actually clip this tetrapeptide bridge. So bacteria, if they don't have the cell wall, they can't protect themselves and they end up lysing. So we've figured out a way for us to create antibiotics that are going to go in and actually break these tetrapeptide bridges, breaking up the structure of the cell walls. So here's our gram positive. And again, this is just kind of a cross section, excuse me. This is a cross section of this cell envelope structure. So this whole entire thing, as you can see here, is called the envelope. It's that series of membranes. This innermost membrane, which we'll get into in a few moments, is called the cell membrane. And you'll notice these little phospholipid here that make up that cell membrane. And then this big brown block that you see sitting on top of it here, this is the cell wall. This is the peptidoglycan right here. And remember, the reason why this appears like this pictorially is we said that this cell wall is extremely thick. There's multiple layers of peptidoglycan in the gram positives. You'll also notice that these two membrane layers are not sitting on top of each other. If you look really carefully, there is actually a very narrow space in between this cell wall and this membrane, this cell membrane here. We call this the periplasmic space. As things move in and out of the cell, this is where a lot of the reactions occur for those materials in this periplasmic space. The final piece which I want to point out is these little strands that you kind of see coming off of the cell wall. These are what we call tacoic acids and lipotacoic acids. I want to be specific in that gram positives are the only cell type that contain the tacoic and lipotacoic acids. Gram negatives do not possess these. So we already mentioned it's a very thick layer of peptidoglycan when we're talking about the gram positive cell wall. Unlike the gram negatives, they include this tacoic and lipotacoic acid, which is going to help for not only maintenance of the cell wall, but also for things like enlargement during cell division. They help to move cations, positively charged molecules, across the cell envelope, and they can also stimulate an immune response. So they do have multiple functions in terms of a maintenance role for the gram positive cell. And we mentioned that there is also between the cell wall and the cell membrane, there is this very narrow space, which is called the periplasmic space. On the other side of that, if we look at the gram negatives, this one's a little more complex. You'll notice now we have a lot more stuff going on here. So as I mentioned to you earlier, there's actually 3 layers with a gram negative cell. We have this outer membrane, which you see sitting on the top, and it's kind of this hybrid of these green molecules here and these beige molecules here. Then if you look really carefully, you'll notice that we also have the cell wall, but this block is much thinner now. It's much less layers of peptidoglycan in the gram negative, followed by the cell membrane, which sits closest to the interior of the cell. So down here in this white space, you can think of this as the interior or the cytoplasm. So we're starting from the outside, so this would be the external environment out here, all the way down to the interior or cytoplasm of the cell. So we already mentioned that gram negatives also have a periplasmic space. It's a little more pronounced than the gram positives, where it was very narrow. The gram negative periplasmic space is much more larger. There's greater space in between those membrane layers. And we mentioned that they have the additional layer known as the outer membrane. And the outer membrane contains a material known as lipopolysaccharide, and it's abbreviated as LPS. We're going to see that one of the most important things about this outer membrane is when the gram negative cell breaks up. So if the gram negative cell is destroyed, the lipid portion of this LPS layer is actually a toxin. We call this an endotoxin. So when that outer membrane fragments, that LPS can actually be a toxic effect to host cells. The outer membrane also can function as a receptor site for things to bind to and enter the cell. And they actually contain these little holes at the top known as porins. And these porins allow for molecules to enter and leave the cell. So it's another way for materials to be transported in and out of the cell. So again, just in summary, gram-positives, 2 layers, the cell wall, which is a thick peptidoglycan layer with these embedded tacoic and lipotacoic acids responsible for maintenance. And then the cell membrane, which is the most internal membrane with a very narrow periplasmic space. Gram-negatives, 3 layers, the outer membrane, followed by the peptidoglycan cell wall, very much more pronounced periplasmic space, and then the innermost cell membrane. So we do have some similarities between the two, but we also do have some key differences. So this is table 4.1 out of your text. Again, a nice little reference tool as you begin to study and read the chapter that gives some comparison to the things that we just went over. So it's important for us to come back. We talked about in chapter 3 some of the different staining techniques that we use to increase the contrast of cells to be able to visualize them under the microscope. So we're going to review and return to the gram stain for a moment. And remember that this was a differential stain. Differential meaning it allows us to distinguish between multiple cell types. The gram stain occurs because of these differences in the peptidoglycan cell wall. So we mentioned that the gram-positive have a really thick peptidoglycan layer. It's going to act almost like a sponge. That gram-positive cell is going to soak up the first stain we call the primary stain, this crystal violet, and it's going to cause gram-positives to stain purple. So think about putting a dry sponge in a bowl of purple dyed liquid. That sponge is going to soak up and pull in all of that purple liquid and get this kind of purple color to it. That's why gram-positives, when we stain them, they're going to stain purple in color. Gram-negatives, on the other hand, that peptidoglycan layer is so thin that we can actually dehydrate it very easily and remove that crystal violet. So no matter what the cell is, it's initially going to take up that crystal violet. Gram-negatives, since their cell wall is so thin, it's going to be very easy to remove the crystal violet from that thin peptidoglycan layer. So they are going to, once they lose that crystal violet, when we add the counter stain, which in this case is called safranin, those cells are going to stain pink in color. So the reason why this is differential is if we're looking at a mixed slide with both gram-positive and gram-negative cells, we should see some purple and some pink cells on the same slide. So we're able to see different cell types based on their cell wall structure. So the major criteria in placing bacteria into groups is based on the structure of their cell walls. That's a really important piece to walk away with from this course. That we start with initially putting bacteria, classifying them in groups based on the structure of their cell wall. So we've already kind of talked about the effects of the gram stain and the different stains. Again, and this will walk you through the different steps. So we notice here's our primary dye, crystal violet, is added. Both cell types are going to turn purple in color. They're both going to soak up that crystal violet like a sponge. Again, gram-positives, much thicker layer. Gram-negatives have this much thinner layer. Then we use what's called a mordant. And a mordant is a fixative agent. It's going to help that dye, that initial dye, to really attach to the cell. We call that grams iodine. So that purple is going to really stick in there. Then we use an alcohol, isopropyl alcohol or ethanol, and we decolorize the cell. Remember we said that this thick layer is going to really hold on to that crystal violet very well. The outer layer of the gram-negatives is going to be weakened, and that dye, that crystal violet, is going to be able to escape. So this is going to go back to being colorless again. So when we add the counterstain, that safranin dye, since the gram-positive is already saturated with crystal violet, it's not going to take up the safranin. But since this one is colorless, this gram-negative cell is going to soak up all of that pink dye, and thus we're going to get the differential appearance of gram-positives being purple, gram-negatives being pink. Now, I mentioned to you earlier, we also have the other category. And the other category are what we call non-typical cell walls. So in the case of Mycobacterium and Nocardia, these have a gram-positive cell wall structure, but they have a high degree of this lipids, waxy material called mycolic acid. And this presents a high degree of resistance for those cells. It's very difficult to stain and destain them because that lipid layer presents an extra barrier that the dye has to get through. And we have cells that lack a cell wall altogether, so they don't even have a cell wall. And this particular group is what we call the mycoplasma. So be careful. Again, it's very similar. Mycobacterium have a cell wall structure. They have this increased amount of lipid called mycolic acid. The mycoplasma, which causes a condition known as atypical pneumonia, do not have a cell wall at all. They have this sterol structure, which is more similar to what we would find in a fungus. And they're what we call pleomorphic. They can have multiple shapes to them. So morphic meaning shape, pleo meaning several. They can have several different shapes to them. So now we're going to go in and we're going to talk about the innermost layer to the cell envelope. And we call that the cell membrane. And really what I want you to think of when you think about the cell membrane is this is a very fluid layer. So this layer is constantly moving. It looks almost like waves of the ocean. So this is going to go up and down. It's going to undulate. So we actually call this, these are proteins here, and then we have these little phospholipids, which look like these little balloons on a string. They actually refer to this layer as proteins afloat in this sea of phospholipids. So this is a very fluid layer. It's not static. It's not sitting still. And the cell membrane carries with it several important functions for the cell. It's going to provide a site not only for chemical reactions to occur, but it's going to allow for passage of materials into and out of the cell. So you'll notice this one here, we have a transport protein with a channel in the center of it. That's going to allow for materials to enter and exit the cell. And the cell membrane really needs to be what we call selectively permeable, meaning that it's going to decide what's allowed into the cell and what's pushed out of the cell. So it's going to carefully decide what's allowed to come in. We call this selectively permeable. And then finally, inside the cell, we've already talked about the cytoplasm, which is this gelatinous material that consists of things like sugars and amino acids. But we said about 70% of the cytoplasm is comprised of water and drives a lot of the chemical reactions that occur within the cell. And then again, since we're talking about the prokaryotes, they don't have a nucleus. There's no membrane-bound organelles in a prokaryote. So they actually have a region where their genome, their genetic material, is located. And we call this the nucleoid region. And in the nucleoid region, we can find two different materials. We find the regular DNA chromosome, which is this circular double-stranded DNA molecule that contains all of the genetic blueprints for that particular cell. In some cases, you will also find these other very small circular DNA molecules called plasmids. And plasmids do not have any effect on the essential capabilities of a bacterial cell, such as growth and metabolism. They often carry excess genes, and they can often be they can enter a cell from infection from a virus or other type of microbial agent. We have found a way in genetic engineering to use plasmids to move genetic information from one cell to another because they're really easy to copy. They're really easy to cut and add particular genes onto, and then to be able to transfer from one cell to another. So we use plasmids quite frequently in genetic engineering to do all sorts of types of experiments and create genetically modified organisms. And then we have the ribosome. We mentioned earlier that the ribosome are found in the cytoplasm, and they differ in their size and the number of proteins from eukaryotes. And ribosomes, key thing, they are the site of protein synthesis, and they are found in all bacterial cells. If they do not have ribosomes, they cannot execute the creation of proteins, and thus they will not be able to survive. The other internal structures that we'll see are these bodies known as inclusions and granules. And the inclusion bodies are designed to be storage areas. Many bacteria will either use them to store water. We have bacteria that actually have a degree of buoyancy because they'll store gases in them, and that allows them to float. Or they'll store nutrients within them or water within them. If they are, in a really dry environment, they'll actually take in water and store it intercellularly. And bacterial cells use them if the environmental nutrient sources get depleted. So these inclusion bodies are really important for survival. The cytoskeleton is this internal network of protein that is basically like scaffolds that support the cell wall. So they are closely associated with the cell wall. They have some degree where they support movement, but they support the structure of the cell itself. So think of it as kind of the beams in your house that support your roof from not falling down. They have a job of maintaining some of that structural integrity of the cell. And then we also talked about endospores. And endospores are these structures that are produced when bacteria are exposed to stress conditions in the environment. And there are 3 gram-positive genera or genus of bacteria that can form these. We have the Clostridium, the Bacillus, and a third genus, which is known as Sporosaxina. So Clostridium, Bacillus, and Sporosaxina are the 3 genera that are able to produce these kind of inactive states under stress conditions. So endospores have a 2-phase life cycle to them. When the cell is active and it's growing and it's in its rod-shaped appearance, we call this the vegetative cell. This is the happy cell. It's moving around, it's taking in nutrients, it's reproducing, it's metabolically active, it's growing. However, let's say all of a sudden I expose those cells to a source of UV light. Now, we talked about back in chapter 1, when these cells, these Clostridium, these Bacillus, the Sporosaxina, are subjected to a stress condition, they can almost do like the turtle does. They can pull their head and arms into the shell. And when they do that, they actually go dormant. They protect themselves. So when these vegetative cells are exposed to stress conditions, nutrient depletion, harmful chemicals, UV light, they can go into and create this dormant structure that's capable of surviving for very long periods of time in these very stressful environments. And they surround themselves with this protein coat, which is a material called keratin. It's the same tough material that we find not only in human skin, but our nails as well. So they surround themselves in this keratin coat, and they can stay like that until the stressor has been removed, and then they're able to return to that vegetative state. So, two definitions for you. Sporulation is the process by which endospores form. Germination is when the spore comes out of its endospore state and goes back to the vegetative cell, actively growing metabolic cell. So, if it's going from a vegetative cell to a spore, we call that sporulation. Draw yourself a diagram that will help you kind of remember which way the terms go. Germination, on the other hand, is when the spore goes back to the vegetative cell. Think about it this way, one of the easiest ways. Think sporulation, the beginning of this word spore. It's forming spores. Germination, when flowers grow, they germinate. So germination means to be actively growing. So they're going back to this vegetative state. So I try to give you little tricks to help you remember some of the terminology. I'm not going to get too much into this. You do not need to memorize the cycle. Do not go crazy trying to memorize the steps. Again, I just want you to understand sporulation is the process where we go from this actively growing vegetative cell to the endospore. We mentioned again that endospores are inactive. They have this thick keratin coat on them, and they can survive for extremely long periods of time. There's been ones that they've known that have survived for 250 million years. And they are one of the most resistant forms of microbial life to all sorts of treatments. So they are resistant to ordinary cleaning methods and boiling. They really have to be subjected to very high levels of heat and steam. We use a device called an autoclave, which is basically a giant oven that exposes the cells to high steam, usually about 20 psi for 120 degrees Celsius, very high temperature, for a long period of time, about a half hour, to destroy the spores. So extremely resistant. All right, we are going to transition and start talking about shapes, arrangements, and sizes of bacterial cells. So there are three basic categories of shapes for bacterial cells. We have the coccus, which is round or spherical. We have the bacillus, which is cylindrical or rod-shaped. Within this bacillus category, we have two kind of subcategories. We have the coccobacillus, which is kind of a shortened rod, and it has a plump ending to it. So it kind of almost looks like, instead of a circle, more like an oval. And then we have a Vibrio, which is kind of comma or kidney bean shaped. It has this curve to it. So it's a curved rod. And then the third category is what we know as the Spherulum, which are these twisted kind of helical rods. And the one subcategory under here that we talk about a lot is the Spirochete, which has this almost spring-looking appearance to it. Looks like like you would see a spring. So it's got this really twisted shape to it. So here's some examples. Here's our coccus, our nice spherical shaped. Then we have our bacillus, which is our cylindrical rod shape. Here's our Vibrio, which kind of has this curved comma or kidney bean shaped appearance. Here's our spirochetes, which have this kind of wavy looking appearance, this almost like a spring looking shape to them. And then we also have ones that are known as Spherulum, which kind of have this wavy-like appearance as well. So lots of different shapes that we're going to see as we go through the chapters this semester. Some microbes can be what we call pleomorphic, and that means that they can vary their cell shape and size. So they can change their outer appearance. Now, we not only have the shapes, bacillus, coccus, and spiral, but we can also arrange these in different manners. So when we're talking about the cocci or the spherical shape, they can either be single, they can be what we call diplo or in pairs, they can be in tetrads or groups of 4. We can form clusters, which we call staphylo. So next to clusters on your slide, please write staphylo. They can be in chains. So I want you to write the word strepto, strepto. Or they can be in these kind of cube shapes, which we call succina. Here's an example right here of a succina, this cube-shaped appearance. Bacilli or rods, on the other hand, can be diplo in pairs. They can be chains, which we call, again, strepto. And they can also be in this palisade or almost like a picket fence appearance to them. So how do we classify prokaryotes? Well, there's 5 different parameters, and this is very similar to what we've talked about previously so far in the course, based on their appearance under the microscope, based on the appearance of their colonies by the naked eye, looking at it with your eyes, by their physiology, so what they do in terms of their nutrients and their reproductive capabilities. We can look at the serology or interaction with immune cells, and we can also study their genetics. And we use, and I mentioned this before in one of our previous chapters, we can use a reference book known as Bergey's Manual, and it's a 5-volume series that contains the information for all known prokaryotes broken out into 2 domains, the archaea and the bacteria. So we're able to go in and refer to and look at the known information for any bacteria to compare it versus an unknown microbe that we're attempting to classify. So again, we mentioned 2 major groups of prokaryotes. We have the archaea, which live in the extreme habitats, and the bacteria. And again, don't go too crazy. I'm not going to ask you a lot on these, the phylums here. I just want you to know specifically that we have the 2 domains, bacteria and archaea, that make up the prokaryote. In terms of the diagnostics, oftentimes we use phenotypic. So whenever you hear the word phenotypic means physical appearance. So we use these physical qualities to identify microbes. So we may look at the cell wall structure. We may look at how they're arranged, what their shape is, what their physiological traits are. And that helps us right away in the medical setting to be able to try to classify microbes that are unknown to us. Again, I'm not going to ask you, this is just an overview of some of the genera that we're going to talk about this semester. Right now, I am not going to ask you this information. As we start to study it and get into it, we will look specifically at a lot of these different families that you see in here. We will go through pretty much every one of these this semester. So this is just an overview for now. If you're interested in learning a little more, this is table 4.4 in your textbook. We also want to talk about the difference between species, strain, and type, because you will hear these words. So starting with the most general, species is a collection of bacterial cells that share a similar set of traits. A strain, on the other hand, differs in that from its parent, there's changes to its metabolism or other cultures of that species. So a strain differs a little bit from its parent species. Okay, so it may have a few subtle differences to it. And then type is what we know as a subspecies that has very key differences in its antigenic makeup. So a lot of times the bacteria will actually display molecules on the surface of the cell to allow it to attach to a host. So anytime there's changes to this antigenic makeup, we would then have to call this a different type. We also have several prokaryotes that have exhibited very unusual characteristics. So we talk about, we have several microbes that are free living that don't cause disease. So they just they're out in the natural environment. They really don't play any immediate positive or negative role. They're just out there living within nature. We also have several bacteria that have photosynthetic capabilities to them. So they utilize light to create their energy. So there's three big groups that we have. We have the cyanobacteria, and the cyanobacteria are important because these are the earliest photosynthetic microorganisms that we know of, and they were formerly called the blue-green algae. The cyanobacteria are what we call oxygenic phototrophs. The reason why they're called oxygenic is because they generate oxygen during the process of photosynthesis. So one of the byproducts they release is oxygen. And they're called phototrophs because they utilize light as their energy source. We also have what's called the green and purple sulfur bacteria. And these are a little bit different in that these are anoxygenic photosynthesizers. Anoxygenic means that they do not derive oxygen as a byproduct when they undergo photosynthesis. The purple and green sulfur bacteria, as you can see by the word sulfur, actually use hydrogen sulfide as their source of electrons. So that's really important. That's a key difference. The green and purple sulfur bacteria are anoxygenic. They do not generate oxygen as a byproduct, and they use a chemical compound called hydrogen sulfide as their source of electrons. And then we also have the gliding or fruiting bacteria, which also are able to undergo photosynthesis as well. So again, I'm not going to go too crazy here. Cyanobacteria have gram-negative cell walls. They also have this green appearance to them, and that's from the presence of this pigment molecule called chlorophyll that allows them to take in the light energy and convert it into other compounds. We talked about the green and purple sulfur bacteria, which we said are anoxygenic. They do not give off oxygen during photosynthesis. And they contain a pigment as well, which is known as bacteriochlorophyll. And then we have the gliding and fruiting bacteria, which they're called that because they glide over most surfaces, and they are gram-negative. Now, we also have some unusual forms of medically significant bacteria. So these are obligate intracellular parasites, and as we remember from previous chapters, this means that they require a host cell for their survival to live off of. We have the rickettsias, which are these tiny gram-negative bacteria. And one of the most common ones that we'll talk about this semester causes a condition known as Rocky Mountain spotted fever, and it's called Rickettsia rickettsii. So again, remember, anytime we write the genus and species name, we always capitalize the genus name, lowercase the species name, and we italicize both names. We also have the chlamydias, which two of the common ones that we'll talk about this semester are chlamydia trachomatis, which causes a eye infection to the conjunctiva and is one of the most common STIs that we know of, and chlamydia pneumoniae, which causes significant lung infection. Now, to wrap us up today, we're going to get into the final group of prokaryotes. We're going to talk about the archaea. And this is remember, 3 domains. This is the third domain. So we have the eukarya, which are the eukaryotes. Domain 2 is the bacteria, which we've talked about mostly today. And the third domain are our archaea. And the archaea are more closely related, believe it or not, to the eukaryotes than they are to the bacteria, even though they are prokaryotes. They contain very unique genetic structures within their ribosomal RNA. And we already mentioned that their cell wall is different from bacteria. They have a molecule called pseudomurine in their cell walls, unlike the bacteria, which have peptidoglycan. This is just kind of an overview. This is table 4 or 5 in your book that compares the 3 domains. So let's kind of run through some of this. So as you can see, the bacteria and the archaea are both prokaryotes, whereas the eukarya are eukaryotes. Bacteria and archaea both have these small circular chromosomes, whereas eukarya have several linear chromosomes. The bacteria and the archaea have 70S ribosome sizes. The eukarya has what they call an 80S. But the archaea, very similar to the eukarya, even though they have 70S, their structure is much more similar to the eukaryotic ribosome. Archaea do not have this peptidoglycan in the cell wall. They have another molecule known as pseudomurine. They have synthesis similar to the eukaryotes, and their cell membranes actually have different linkages from the bacteria that can be much more similar to the eukaryotes. So we mentioned that the archaea live in these very extreme habitats. So they have adapted themselves to places like high heat. We call those thermophiles, or high salt concentrations. They've been known to live in the Dead Sea, which has some of the highest salt concentration on Earth. We call these halophiles. They can live in acidic pHs, very low pHs. We call those acidophiles. And they can even survive in high pressure in atmosphere. We call these barophiles. They can survive in very high pressure environments, like the bottom of the ocean. We also have in this group, so we've talked about things like the hyperthermophiles, which survive high heat, the halophiles, which survive high salt. We also have these methane producers, which we call methanogens. And what they do is they oxidize hydrogen gas. And when they oxidize this hydrogen gas, they produce methane. Even though the methanogens or methane producers are in the archaea group, they really are not extremophiles. They're not living in these really intolerable environments. They're classified here because they're able to take something like hydrogen gas and convert it to methane, which is very unique and unlike anything we've seen with the bacteria. So that is the end of our discussion for today, the end of our talk. Again, if you have any questions as you're reading, as you're looking through the chapter, please feel free to reach out. Let me know during office hours or on the questions for the instructor in our discussion board. I look forward to seeing everybody soon. Hope everyone's doing well, and take care.